Hello, welcome back to Intro to Biology for Majors, or Biol 1224. This PowerPoint is going to cover chapters 14 and 15 of your Openstax book, Biology 2E. This is in the third module of this course, and chapters 14 and 15 covered DNA structure and function and genes and proteins. So as always, we're starting off with our objectives. So the first is recognize DNA structure and mechanisms. The second is identify 3 major replication enzymes and their function. The third is recognize the central dogma components, location, and steps. And the fourth is describe the significance of DNA mutations. So let's start off by talking about DNA. So some DNA facts are that DNA stores the information that the cell needs to produce proteins. Mitosis actually requires 2 copies of DNA. DNA is composed of nucleotides, which we've talked about before. They are adenine, which is represented by an A, and it binds to thymine, which is represented by a T, and they're guanine, which is represented by a G, which binds to cytosine, which is represented by a C. So hydrogen bonds, the weak bonds that we've talked about before, actually connect complementary DNA strands. So once again, A to T and C to G, like rungs on a ladder. And the backbone, which would be in this image, the blue portion, which you can actually see right here. This is the backbone they're talking about. That's held together by covalent bonds, which are stronger bonds. And then once again, these complementary strands of DNA are held together by hydrogen bonds. So a few of the notes that I have here that I'd like to kind of go over. Once again, DNA is a double-stranded molecule composed of 2 separate RNA molecules that forms a double helix by binding A to T and C to G. Base pairs like C and G, once again, are bound together by hydrogen bonds. The ribosomal subunits join, allowing mRNA, which is mitochondrial RNA, to move through the ribosome. Each tRNA contributes its corresponding amino acid, allowing the polypeptide chain to form. When translation is complete, the components disassemble. So each DNA nucleotide is made up of 1 5-carbon sugar, a phosphate group containing a phosphate, and a nitrogenous or nitrogen-containing base. So that's each different nucleotide. So the A and the T and the G and the C are all composed of a 5-carbon sugar, a phosphate group, and a nitrogenous base. All of an organism's genetic material is known as its genome. And once again, DNA is composed of nucleotides, being A, adenine, T, thymine, G, guanine, and C, cytosine. Okay, so let's talk about the process of replicating DNA, which means making more DNA. So helicase is what we call an enzyme, and we've talked about enzymes before in previous PowerPoints. Enzymes act as the catalyst for reactions. So a catalyst means forcing something to start, right, or beginning something. So helicase is actually what unwinds a DNA molecule here. So if you look on the left side, you can see we still have a DNA strand that is still within the double helix, working as it normally would. And if you look right here, this yellow arrow that's pushed the DNA apart and made it 2 separate strands rather than 1 combined double helix, this yellow arrow is your helicase. So it's pushing that DNA apart, pulling it apart, and unwinds. So then we have something called DNA polymerase, which you can see here and up here. DNA polymerase actually comes in and it creates a complementary strand. So if you're looking, the actual strands that are left behind from the original DNA are this bottom strand and then this very top strand up here. So the DNA polymerase is coming in and making this complementary strand for the top and this complementary strand for the bottom. So it's coming in and saying, okay, we have an A and a T, so we're going to have a T and an A down here. We have a G and a C, so we're going to have a C and a G down here. So it's actually matching up and creating a perfectly complementary strand to the original. And then we have the enzyme ligase, which you can see right here. Ligase actually forms covalent bonds between the adjacent matching nucleotides, which are strong bonds, and it allows the bonds to actually form together within those nucleotides. So that's what ligase is doing in this image. So a few of my notes here that I have are replication and transcription actually rely on DNA that is found in the nucleus. So translation occurs at the ribosome, and meiosis forms what we call gametes. Later on, we'll learn more about that. But translation is when you're basically reading that DNA. That occurs at the ribosome. So the semi-conservative method suggests that each of the 2 parental DNA strands will act as a template for the new DNA to be synthesized. So then after replication takes place, each double-stranded DNA includes 1 parental or old strand and 1 new strand. So we can actually see that in this image where we have the parental strand, which would be that old strand on the very bottom that's from the original DNA, and then the old strand from the very top up here. And then we have the new strands that DNA polymerase put together. So we have the matching on the bottom here, the matching on the top here, and then ligase is going to form those covalent bonds to get them to actually come together. Okay, so let's talk a little bit more about replication. So we're still hitting really big on DNA in these slides. We're going to move on to RNA, transcription, and translation later on, but we do kind of talk about those things here as well. So replication actually doubles a cell's DNA. So if you look at this image here, this is the original parent DNA. We're going through the process of replication, and then we have the red strand on this side, the blue strand on this side that were the original parent DNA. And we have newly synthesized strands of what we call daughter DNA, and now we have 2 separate daughter DNA strands or daughter helices. So replication doubles a cell's DNA, and after replication, each DNA molecule has 1 parental strand like we talked about in original, and then they have 1 new daughter strand. So DNA codes for a protein via the process of transcription, and then the protein is coded through RNA, which is done through translation. So we're going to keep kind of hitting on that in the next few slides, but that's just introducing the topic. So there is one organelle that we can actually see with the light microscope, which we did see in our lab. So you can actually see the nucleus, which contains chromosomes, DNA structures with specific genes that code for specific proteins. And during the process of cell division, which we're going to hit on in the next few slides, each one of these daughter strands goes to a daughter cell. So when we have an original cell and it decides it needs to replicate, it needs to split in 2, we have to then replicate our DNA as well, so that each new cell has a new double helix of full DNA. So that's DNA replication. So the process of transcription also occurs in the nucleus, and it yields an mRNA, which we call a messenger RNA. And then translation will occur in the ribosome, and it yields a polypeptide or a protein. So we're going to hit more on that in the next few slides. We're going to talk about the central dogma and kind of get more into that, but this is just introducing the topic to you. So now let's talk a little bit about what we call the central dogma of this process. So this is going to describe the genetic flow of information in the cell. So this is one of our objectives, so make sure you're paying attention. So the central dogma, if you look at this little indicator here that I have, it goes from DNA to RNA to protein. So protein production starts with DNA. A gene is a sequence of DNA that encodes a specific protein. So protein production occurs in 2 different stages. So we have transcription and we have translation. So we're going to get into the next few slides where we actually talk about RNA. We've already talked a lot about DNA. We're going to talk about RNA, and we're going to talk about transcription and translation as well. So once again, the central dogma describes the flow of genetic information in the cell from genes to mRNA to proteins. So we would call it genes or DNA to mRNA to proteins. Genes are used to make mRNA by the process of transcription. So DNA starts, and then we have this arrow here, which would represent transcription, going to RNA. And then RNA is used to synthesize proteins by the process of translation. So this blue arrow would represent translation, making a protein. Almost every species on the planet uses the same type of genetic code, and we go through this same kind of process. So DNA uses transcription to make RNA, which uses translation to make a protein. Okay, so let's talk more about RNA. So there are 3 types of RNA that interact to produce proteins. So the first one we've seen a little bit in the previous slides is messenger RNA. So mRNA, messenger RNA carries information from the nucleus to the ribosome. So then we actually have ribosomal RNA, or rRNA, and we also have what we call transfer RNA, or tRNA. And a transfer RNA will carry amino acids to the ribosome. So RNA, unlike DNA, contains adenine, guanine, cytosine, and what we call uracil. So rather than thymine, there is no thymine. We have A and U and G and C that match up. So those are the nucleotides we talk about in RNA. RNA is a single-stranded molecule composed of adenine and uracil, cytosine and guanine. So if you look here at the image, you can see that it's basically 1/2 of a DNA molecule. So it's just one of those two strands that would be considered RNA. And then here we talk about a ribosome. This is where you can see ribosomal RNA. And then when we talk about an amino acid up here, we have transfer RNA down here. So this is just kind of showing you an image of what the different types of RNA look like and where they belong. Okay, so now let's talk a little bit more about transcription. So transcription occurs in the nucleus. The DNA template strand encodes the RNA molecule. The RNA then leaves the nucleus to be translated. So whenever we talk about transcription, we're talking about a process in the nucleus where the DNA template strand, the original strand, encodes the RNA molecule. So if you're looking at the image I have right here, we have the original DNA strand up here, and then we have the process of transcription where this original strand codes for an individual RNA molecule. So like I said, this RNA is basically just half of that original parent DNA here. So we're creating what we call an mRNA or a messenger RNA strand that's then going to leave the nucleus to be translated later on. So we're transcribing it from that original DNA, we're going through transcription, and then we're going to have this new RNA that's a messenger RNA that'll then leave to be translated. Okay, so now let's talk about translation. So this just continues a very similar image, but in translation, it converts the messenger RNA message that was sent into an amino acid sequence. So we have the original RNA, the mRNA we got from the last image, so it went from that original DNA strand to this new mRNA, and then we translate and we create a protein. So we go from DNA in the last slide to mRNA or RNA here, and then we make a protein. So that's that central dogma that we talked about initially. So during the process of translation, the mRNA template provides specific information in units of 3 nucleotides, which we call codons. So we literally will have 3 nucleotides that code for this protein at a time. And we call those 3 nucleotides together codons. And there's going to be an image in the next slide that kind of shows you what I'm talking about even more. So translation converts the mRNA, the messenger RNA, into what we call an amino acid sequence once again. And that would then create your protein. So let's move on to the next slide, which really kind of shows you an image and digs even more into that process. Okay, so the process of translation builds the protein. And we've talked about proteins before. Proteins do all the work, right? It's their job to do the work in our bodies. So translation builds the protein. A codon, which we talked about in the last slide, is a 3 nucleotide sequence that encodes 1 amino acid. So if we're talking about a codon, see right here, we actually have 3 on the bottom here on this mRNA strand. We have 3 nucleotides. We have A, C, G. So those nucleotides would then encode 1 amino acid. And so in this process, we have translator RNA, we have the tRNA, which is going to see this attachment site to this codon. These are going to be the subsequent or the matching nucleotides with the tRNA that code for this specific amino acid. And of course, this is not exactly how it looks. We're just kind of giving you an image of exactly what I'm talking about to help, visualize. But you have a codon, so a series of 3 nucleotides here that match with your tRNA. There's an attachment site where they kind of get the information they need from the messenger RNA down here, and then they code for this specific amino acid that we need. So the genetic code will actually show which mRNA codons correspond to which amino acid. So then tRNA, what we talked about right here, this big yellow piece, will bind to both the mRNA codon, which is down here, and the corresponding amino acid up here. Transcription and translation in bacteria are actually much simpler, but they do still take place. So this is the process. Basically, this tRNA will come in. It sees the original codon from the messenger RNA, and it takes that message through this attachment site and forms or codes this exact amino acid. And that takes place all the way down. So it'll move from this codon to this codon to this codon to this codon, and through that process, we're making these amino acids that then will make up our new protein that we want. So, protein synthesis, or the process of creating a protein, is actually highly regulated. So protein synthesis requires a lot of energy. It requires a lot of that ATP that our body works so hard to make. So cells save that energy by only producing the needed proteins, only producing the things we actually need to function. So both prokaryotes, or simpler organisms like bacteria, and eukaryotes, which would be plants and animals and the more complex organisms, they all regulate protein synthesis, but in different ways. So if lactose is absent, lactose digesting enzymes are then not needed, and a repressor protein will block the process. These are just examples. If lactose is present, a repressor binds to lactose, changes the shape, and then releases an operator. And protein synthesis of lactose digesting enzymes will then occur. So this is just an example of how it's highly regulated. So in this image here, that's exactly what I'm showing you. I'm showing the kind of the process we talked about before. So we have a DNA strand being broken down by transcription to mRNA, which then goes through, creates this messenger RNA. This all takes place in the nucleus. We transport to the cytoplasm for protein synthesis, and then we have the whole process of protein synthesis happening that we talked about previously here. Okay, so now let's talk about 4 mutation types. So there are ways of mutation within this process. So silent has no effect on protein sequence. That's a silent mutation. Missense mutations result in amino acid substitutions. So basically, missense would mean that an amino acid would come in and substitute where it isn't supposed to. A nonsense mutation will take place. It substitutes a stop codon for an amino acid. So rather than actually producing the amino acid needed, it'll code for the actual translator process to stop. So that's what we call a nonsense mutation. And then we have what we call a frame shift mutation, which inserts or deletes nucleotides, which may result in a shift in the reading frame or insertion of a stop codon. So basically, we'll either insert or delete nucleotides from that original messenger RNA, and this is the mutation. It deletes those, and then the result is a shift, or basically the inability to read the frame or insertion of a stop codon. So it'll either make the tRNA unable to read the original message, or it will stop the whole process. So I'm going to kind of sum that up with my notes. It says a silent mutation has no effect on the protein sequence at all. Inserting or deleting a codon, which with the 3 nucleotides we talked about, is often less harmful than a mutation that involves 1 nucleotide. And a change in 1 nucleotide can frame shift, which impacts the primary codon and every codon downstream. So in the frame shift process, it will affect that primary codon, or those primary 3 nucleotides with the frame shift, but it'll continue to affect the entire thing downshift. So the whole way that you're going, the rest of that translation process is being negatively affected by the frame shift. So those are 4 different mutations that we'd like for you to know here. So mutations can change your DNA. So a single base substitution in a hemoglobin gene causes blood cells to form abnormally, which will lead to sickle cell disease. So that's a form of mutation that we actually know about in real life. So once again, a single base substitution, so a single base pair substituting incorrectly within the hemoglobin gene causes blood cells to form abnormally. They look more like banana shaped rather than circular. And it leads to what we call sickle cell disease. So mutations are not always harmful. They're not always a bad thing. Mutations create different versions of alleles, which are alternative versions of the same gene. Plant breeders even introduce mutations to create new varieties of plants. So for instance, our crops, we actually have hardier crops today than we used to 100 years ago, because farmers and scientists alike have all worked with plants and created new mutations to make our plants more hardy than they used to be. So cancer is best defined as what we call an accumulation of different mutations. A tumor is an abnormal mass of tissue, and that's from this accumulation of mutations. The cells of a tumor divide out of control, and tumors are either benign or malignant. So benign meaning basically not harmful, not cancerous, or malignant meaning cancerous. So cancer is literally caused by gene mutation. Or DNA mutation. This process happens naturally, This is something that happens in our bodies all the time, and our bodies are sort of able to move past it because it's a small change within our cell cycle. But if we're talking about things like sickle cell, cancer, we can have harmful cell mutations, DNA mutations. But then if we talk about breeding plants and things like that, we can have positive DNA mutations. Okay, so we are to our key takeaways from these chapters. So, 1, the DNA has 2 complementary strands composed of A, C, T, and G, which would be adenine, cytosine, thymine, and guanine. And then we talked about our enzymes helicase. So helicase will unwind DNA. DNA polymerase will add nucleotides in, and then ligase will bind the strands together. DNA goes through transcription with RNA, goes through translation to make a protein. So the first arrow, once again, is transcription in the nucleus. The second is translation, which happens in the ribosome. And the fourth and final key takeaway is that mutations can be helpful or harmful, and one nucleotide alone can cause them. And once again, cancer is an accumulation of mutations. So once again, if you guys have any questions about this chapter or chapters 14 or 15, please just follow up with me over email, and you guys can go ahead and move on to your next chapter.